Ok. Ok, pessoal, vamos dar início a essa manhã. Eu vou misturar aí o português e o inglês, né? Na medida que eu venho a falar com a audiência e, ao mesmo tempo, com o professor Ziff. Well, today we're extremely pleased to welcome Professor Edward Ziff. Edward has been an important collaborator and a friend since his first contact with UFF, which took place through the Professor Roberto Paz de Carvalho in 2004, if I remember correctly. Since then, Ziff has received several students, postdoc professors in his laboratory in NYU, New York University, and was crucial for our scientific production for several collaborative works. And a special feature that I have to mention here, and I noticed when I was in Ziff's lab in 2013, more or less, I can notice that students that love the work of him, and I had a good time with him. And I will start to read the mini resume, o currículo of Ziff, e eu vou ler agora em português, para que todo mundo tenha uma noção, né, do que é o nosso querido amigo e professor. Ziff, I will start to read your curriculum, and I give a word for you, okay?
Okay, thank you.
Ziff, Edward Ziff, vou ler em português. O professor Ziff, ele é bacharel em Química pela Universidade de Columbia, ele é PhD em Bioquímica pela Universidade de Princeton, ele foi estudante de pós-doutorado do vencedor do Prêmio Nobel 2 vezes, o Prêmio Nobel, né? 2 vezes, twice.
2 vezes.
Exactly. 2 vezes Prêmio Nobel Fred Sanger, no laboratório MRC, de Biologia Molecular em Cambridge, atuou no corpo docente do Imperial Cancer Research Fund, em Londres, e na Rockefeller University, em Nova York. Ingressou na Escola de Medicina na Universidade de Nova York, onde é professor de Bioquímica e Farmacologia Molecular e Ciências Naturais. Atualmente, o laboratório do Ziff estuda estrutura de sinapses, estrutura da sinapse, da regulação sináptica, incluindo mecanismos de homeostase e recompensa, de regulação da sinapse na doença de Alzheimer. Ele é coautor de livros, artigos para o público em geral, sobre DNA e autismo. E eu gostaria também de destacar que das principais contribuições que o Ziff teve inúmeras contribuições para a ciência, de um modo geral, inúmeras. É infindável o currículo do Ziff. E dessas inúmeras contribuições, ele foi o primeiro a sequenciar o nucleotídeo do DNA, primeiro também a trabalhar com a RNA polimerase 2, foi que descobriu aí a resposta dos genes de resposta imediatas, como a CFOS,
Yep.
E também o ancoramento de receptores AMPA, né? E no tráfego e sua montagem em sinapses. Well, Ziff, it's a great pleasure, it's a great honor to be with you here. And thank you very much for your presence and availability. It's always good to be with you and hear about your research and trajectory. Please be welcome, and you are very grateful.
Thank you, Marcelo. You know, as you mentioned, it was on my first trip to Brazil that I visited UFF and Niterói, and I met you. And that started a very strong and, for me, extremely positive relationship. And in fact, since then, I've been many, many times in Brazil. And I have taken quite a few students in the laboratory. And in fact, what I'm going to speak about today, in part, is the result of Brazilian students' work. Okay. So thank you for this invitation. So now to show my screen, can you see it now?
Yeah.
Is it okay? Hello?
Yes.
Can you see the screen?
Yes.
My phone. Okay. Very good. Thank you. Okay, so I'm going to be speaking to you today about reward. And as you know, one of the most important functions of the brain is to select and then carry out behaviors. And in this selection, reward plays a very important role. In fact, recognizing and responding to rewarding experiences is one of the major functions of the brain. So, what is reward? So, Wolfram Schutz is a person who has worked very extensively in this area. And he says any stimulus, object, event, activity, or situation that has the potential to make us approach and consume it is by definition a reward. So you can see that there, at least one of the aspects of reward is to induce behavior, the behavior to consume. Okay. So what constitutes a reward? So energy-rich food is rewarding, sexual activity, social interactions with friends. Even if you're thirsty, drinking water, it constitutes a reward. And when you consume reward, when you carry out some rewarding act, the part of the brain that responds and makes decisions about your behavior is the striatum. And the striatum works in conjunction with the release of dopamine, which is a marker for reward. So our studies use consumption of sucrose as a representation of reward. So sucrose can be considered to be a natural reward. Cocaine, for example, which later in the talk I'll speak about, works by a similar mechanism in stimulating the brain, but it's obviously not a natural reward. Sucrose has the ability to motivate or to change behavior. So in a kind of a behavioral test that's applied to rats called conditioned place preference, a rat will choose a position in the cage in which it is able to consume sucrose. So this is consistent with it having a motivating capacity. So most of this talk will be concerned with the pathways that sucrose induces in the brain that modifies the brain function, and leads to formation of behavior. And that pathway starts with the sweet taste receptor, which is present in your tongue. And I'll speak about that a bit later. So here's the organization of the talk. I'll speak about how a reward, consumption of sucrose, which is rewarding, regulates the brain's reward pathways, in particular, AMPA receptor trafficking. So you know that brain plasticity depends to a large extent on regulating the trafficking of AMPA receptors to and from synapses. Okay, we'll speak about that quite a bit. So then I'll ask, how could this regulation integrate rewarding stimuli, such as represented by dopamine, integrate that with sensory executive stimuli, such as represented by glutamate. Okay. And at the end, I'll speak very briefly about how the way the brain responds to sucrose is related to addiction, and in particular, cocaine addiction. So the part of the brain that governs the reward response is the striatum. And pathways in the striatum are represented in this slide. So you can see the pathways start with the cortex, in particular the prefrontal cortex, which is involved in making executive decisions. The prefrontal cortex signals through two pathways, one going through neurons that express receptors for dopamine called D1 receptors, and it also responds through neurons expressing D2 receptors, dopamine receptors. So these are called the direct and the indirect pathways. So this is the indirect pathway, which is more complicated, but we'll be speaking today mostly about the direct pathway. And the reason for that is that the direct pathway is the target for dopamine, which activates D1 receptors and medium spiny neurons. We'll be talking quite a bit about that. Okay. So here's another representation of these striatal pathways. And we'll start over here. Mind you, this is what's called a cortical striatal thalamic cortical loop, which is just a complicated way of saying that there is signaling that goes from the cortex to the striatum, which we'll be talking about a lot, which speaks to the thalamus, and that speaks back to the cortex. Okay, so where is that on this diagram? So we start out here with the prefrontal cortex. So as you know, this is the part of the brain that makes executive decisions. It sort of gathers all the information that it can from other parts of the brain and weighs what is important against what is not important, and ultimately chooses a plan of action. So the prefrontal cortex sends excitatory or glutamatergic neurons to the striatum. And here in the putamen, we find critical neurons that are critical for this pathway, and these are medium spiny neurons. So the pathway involves prefrontal cortex release of glutamate onto synapses of medium spiny neurons here, okay? These then signal to a second, these medium spiny neurons are inhibitory, and they signal to a second inhibitory neuron in the circuit, which then signals to thalamus, where it gates signaling that's related to behavior. So what's represented here is the basal state, when no prefrontal cortex activity is being transmitted. However, when the prefrontal cortex becomes active, it stimulates this neuron, an inhibitory neuron, which shuts down a second neuron in the circuit, which is also inhibitory. And when this neuron is shut down, disinhibition takes place, and thalamic transmission commences. So the thalamus speaks to the motor cortex, and the motor cortex enables you to carry out a behavior, such as picking up a cup of coffee or eating a piece of cake. So this is the pathway we're going to be speaking about. And in fact, excuse me, most of the talk will be concerned with neurons at the beginning of the pathway and the synapses which form with prefrontal cortex neurons, and also extremely important, dopaminergic neurons that project from the VTA. So here there is some cooperation between glutamate, released from prefrontal cortex neurons, and dopamine, released from VTA neurons, onto synapses of these medium spiny neurons. And what we'll be speaking about, or what I'll show you, is that these two signals work together to activate this pathway and make it stronger. And this has an influence on transmission to the motor cortex and the formation of behavior. Okay, so that's a critical synapse. So what are medium spiny neurons? These are the major neuron types of the striatum. 95% of the neurons are medium spiny neurons. They're inhibitory, GABAergic, and they express D1 dopamine receptors. Okay. The other medium spiny neurons in this indirect pathway express D2 receptors, but we'll be speaking mostly about the D1 expressing neurons. So here's a synapse onto a medium spiny neuron, and you see it receives a lot of inputs. It receives, as I said, glutamatergic input. It also receives dopaminergic input. It receives GABAergic and acetylcholinergic input as well. And it's got a host of receptors in the synaptic membrane. So this talk is going to focus on these two inputs, the input from glutamate and the input from dopamine. And what I'll try to describe to you is a cooperation between these two signals in regulating the function of this synapse, okay? And this is a representation of such a synapse. Down here would be a medium spiny neuron. Up here is the nerve terminal of a prefrontal cortical neuron. This here is glutamate ready to be released. The medium spiny neuron expresses AMPA receptors in these synapses. It also expresses D1 receptors, which respond to dopamine released from dopaminergic neurons that come from the VTA. Dopamine activates the D1 receptor, which leads to an increase in cyclic AMP. So what we're interested in is the cooperation of these two inputs to medium spiny neurons when a person experiences something rewarding, okay? So to study this, as I said, we have used sucrose as a representative of reward. And in the first experiments, we simply examined what happened when a rat consumes sucrose. So let's talk about the pathway that is stimulated by sucrose. Of course, sucrose stimulates the tongue, and the tongue has receptors for many tastes, but the critical one for sucrose is the sweet taste receptor, just one of these, okay? So the sweet taste receptor is activated by sucrose, and these sensory epithelial neurons, which express sucrose sweet taste receptors, signal to the hypothalamus. And the hypothalamus signals to the ventral tegmental area, the VTA, which I told you projects to several regions of the brain, the prefrontal cortex, hippocampus, and nucleus accumbens. So one aspect of the signaling of sucrose is this pathway, which leads to dopamine release, in particular in nucleus accumbens, okay? So that's shown here. Sucrose activates hypothalamic function, which in turn stimulates VTA. Here's the VTA, which then releases dopamine. the same time, prefrontal cortex activity is driving the release of glutamate. So we're going to try to understand how two signals, dopamine signal and glutamate signal, affect transmission at this synapse. Okay. So what are the consequences for this system of consuming sucrose? So we'll look particularly at synapse structure and function, and especially AMPA receptors, and how AMPA receptors traffic in response to the consumption of sucrose. And we'll also look briefly at behavior. Okay. So to make this analysis, we compare two groups of rats. Okay. One group of rats consumes sucrose. So the sucrose is presented from a bottle, the rat's in a cage, and the sucrose rats consume sucrose 5 minutes a day for 7 days. Okay. There's a control group which is presented with a bottle with water, and they are on the same schedule of consumption. So notice that these animals only get exposed to water or to sucrose briefly for 5 minutes. The rest of the day, they don't. Well, they certainly don't have access to sucrose. Okay? So that protocol is represented here. Here, day 1, day 2, day 3, day 4, day 5, day 6, day 7. One group consuming sucrose and the other group consuming water. So the first measurement we made was behavioral. We asked whether consuming sucrose altered the activity of a rat, and in particular, the total distance that the rat travels in a cage, or the total horizontal activity as it moves up and down. So these are two parameters of motivation. So you can see on the first day that they consume sucrose, the sucrose and the water rats are more or less the same in terms of this activity. Okay. However, if you look at the 6th day of consuming sucrose, you see a very pronounced difference. The sucrose rats run around more in their cage. The total distance traveled is increased, and the horizontal activity, which is their movement up and down, is also increased. So consuming sucrose has an impact on behavior. This is admittedly a very crude measure of behavior, but still I think it's valid. So now let's look at changes which take place in medium spiny neurons after a rat consumes a rewarding substance, sucrose. And in particular, what we'll look at is AMPA receptors that are present in medium spiny neuron synapses. You know that AMPA receptors are tetramers. They're made up of subunits GluA1, 2, 3, and 4. And these subunits can combine in different ways to make different tetramers. These are the functional forms of the AMPA receptor. And you'll see we'll be particularly interested in one of these, which is composed only of GluA1. These have GluA2, GluA3, and GluA4. You'll see that these receptors have special properties, and they're also inserted into the synapse when a rat consumes sucrose. Okay, so what we did was take rats that had consumed sucrose once, twice, 3, 4, 5, 6, 7 times. We sacrificed them, and we analyzed their synapses for AMPA receptor types. Okay. We did this by Western blotting. So we made two preparations. This one is the whole extract of the nucleus accumbens, but this one you could call the synaptic extract. It contains what's called the postsynaptic density, and it's very much enriched for synaptic AMPA receptors. So you can see that if you look at GLUA1, you don't see much if you look at the whole cell fraction, but if you focus in on the AMPA receptors that are present at the synapse, you can see that by day 7, there's a dramatic change in their content. They have much more GLUA1 than the control animals or animals that had received less sucrose. This is specific because we don't see a change in a second AMPA receptor subunit type, which is GLUA2. So this is the first evidence that simply consuming sucrose makes a change in the synapses in the nucleus accumbens. Okay. We wanted to be sure that these changes actually resulted from sucrose and from consumption of this stimulation of the sweet taste receptor. So we took other rats and fed them with saccharin, which is an artificial sweetener, of course, and it stimulates the sweet taste receptor as well. And we can see that two parameters. Well, here's the first parameter is how much do they drink. So the animals prefer sucrose. They also prefer saccharin to water. They consume more sweet things. But over here you see that just as sucrose increases the GLUA1 AMPA receptor subunit at synapses, so does saccharin. So we can be pretty confident that these changes in synapses that we're observing are the consequences of drinking sucrose. We asked whether the synapses change their strength. So this is critical for signaling down that direct pathway. And you can see that two measures of synapse function. The first called mini EPSC amplitude, which is the amount of current traveling through a synapse when it's stimulated. This is increased by sucrose. Then we looked at what's called the mini EPSC frequency, which is the firing rate of one of these medium spiny neurons. And that is also increased by sucrose. So you can see not only does sucrose change the composition of AMPA receptors at these synapses, it also changes their function. So I mentioned that there are several different AMPA receptor types, and one of these is special. It's called the GLUA1 homomer. Okay, it's made up exclusively of GLUA1. These contain GLUA1 and GLUA2. Okay. And GLUA1 homomers have very special properties. The first thing is that they're highly conducting. They're very strong. They emit very large currents. The second is that they are inwardly rectifying, which means for certain reasons that currents can flow into the cell, and however, current flow out of the cell is very small. The third unique thing about these receptors is that they conduct calcium, while most AMPA receptors only conduct sodium. And this rectification, which I mentioned, it results from what's called a polyamine block. So what we've seen is that when a rat consumes sucrose, synapses in a critical pathway change, and the change I'll show you involves the insertion of this special type of AMPA receptor. So a signature of this special type is rectification. That means that the current into the cell through the receptor is large, while the current out of the cell is small. And that's summarized here in what's called the rectification index. So you can see that, and here's the data for that. So a regular AMPA receptor conducts the same way, whether it's conducting inward or it's conducting outward. But the synapses of rats that consume sucrose don't conduct outward as well. And that's summarized in the rectification index, which is accepted as evidence that this special type of receptor is inserted. Okay. So then we asked, does this special type of receptor have any consequence for the behavior of the animal? And I told you that the animals were much more active after they consumed sucrose. Okay, so that's shown here. The sucrose animals have much more horizontal movement than the animals that consumed water. But if you inject spermine, which is a polyamine, into the nucleus accumbens, which contains the synapses we've been talking about, this difference goes away. So not only do the synapses change, but that change has a functional consequence. So now we're going to try to understand how this special type of AMPA receptor gets inserted into the synapse. This diagram shows you some details about the structure of a special part of the AMPA receptor. So as you know, the AMPA receptor has 4 subunits. Each one has what's called a C-terminal domain, or cytoplasmic domain. So for simplicity, I've only shown one of these. And there are a number of structural features in this domain, which I should point out. There are 3 different serines that get phosphorylated, and you'll see that these control the movement of the receptor into the synapse in response to dopamine and glutamate. And there's another sequence here, which I'll speak about later. And in particular, these serines and this sequence control the trafficking of the receptor, the movement into the synapse and out of the synapse. Okay. So I'll be speaking a lot about this particular serine, serine H45, because its phosphorylation is critical for regulating the trafficking. Okay. And I should point out, this is an experiment done in collaboration with Ken Carr, a very good colleague at NYU, that when a rat consumes sucrose, in particular a rat that has been food restricted, it hasn't had sucrose for some period of time, this particular serine H45, becomes phosphorylated. And you'll see that that's phosphorylation, which takes place in response to sucrose, is critical for changing the function of the synapse. So what's known about how AMPA receptors get into synapses? So this picture represents a dendrite, a part of the dendritic arbor of the medium spiny neuron. And this is a spine, which is a projection from the dendrite. And this is it contains the synapse, and this is up here is the postsynaptic membrane. So the AMPA receptors, you'll see, will move up to the synapse. They start out inside the dendrite, and they move by exocytosis into the plasma membrane, and then they can move back again unless something happens. Okay? So receptors that are in the membrane can be phosphorylated by protein kinase A. And this takes place at that important serine 845. And as time goes on, sorry, these should be all white, not red and white. As time goes on, more and more receptors accumulate in the postsynaptic membrane. So mind you, they're not in the synapse yet. They would have to travel here to the postsynaptic membrane to actually reach the synapse. Sorry if there's noise outside. Okay. After they get to this extrasynaptic site, through a second signal that comes from protein kinase C, they enter the synapse. Okay, so keep this pathway in mind. If they get dephosphorylated by the phosphatase calcineurin, they can move out of the synapse and eventually back into the dendrite. So we wanted to know how this pathway operated in rats that had consumed sucrose. And we did this by making sections of rats that had been exposed to sucrose or rats that had drunk water. And we stained these sections with immunogold, and then we looked at them by electron microscopy. And we counted grains, which showed us the location of AMPA receptors in these tissues. So we look for AMPA receptors in 3 locations. One is inside the spine. Here is the spine in this image. And a second location is extrasynaptic, so not yet in the synapse, but traveling to the synapse. And the third is synaptic. Okay? So quantitation of many, many, many electron micrographs gave this result. So let's look first at what happens to animals that have, let's look first at receptors that are at the postsynaptic density. We have 3 groups of rats that we're looking at. Ones that just drank water, ones that drank sucrose for 6 days, but on the last day, they only had water. So they hadn't seen sucrose for 24 hours. And the final group, the third group, has consumed sucrose just 5 minutes before they were sacrificed. Okay. I'm sorry for this noise outside. There's nothing I can do about it. The second group, or the second location we looked at, was intracellular. Oh, let me go back here for a second. You can see that the receptors at the synapse are increased when the animals consume water, and the increase is seen if they've consumed water recently, sucrose recently, or if they've consumed sucrose 24 hours in the past. So we would say that if the receptor can reach this location, it's stable. Okay? The second location is intracellular, and these animals show sucrose if they've show AMPA receptors at the intracellular location, if they have consumed sucrose recently, or even if they have consumed sucrose only 24 hours ago. So this population also goes up and it's stable. But the third population, which is extrasynaptic, behaves differently. If you consume sucrose recently, it's high, but if you consume sucrose a long time ago, or if you've never consumed sucrose, it's low. Okay. So the conclusion from this study is that sucrose induces a stable pool of intracellular receptors, a transient pool of extrasynaptic receptors and a stable pool of synaptic receptors. And in fact, this actually is very similar to the pathway I showed you a moment ago, in which GluA1 moves to an extrasynaptic location and then in a second step moves up to the synapse. Once it gets here, it's stable. But if it only reaches this position, it's transient. In a few minutes after arriving, it can go back into the cell. Okay. So how does this unstable extrasynaptic pool form? So the formation depends on elevating the level of the receptor in the membrane. This can take place by two mechanisms. You can increase exocytosis, which is the delivery of the receptor to the membrane, or you can stabilize it and decrease endocytosis. So we'll try to distinguish between these two possibilities. First, looking at exocytosis. So we do this with what's called a FRAP experiment. We express in cultured neurons GluA1, which is tagged with fluorine. So fluorine is a form of green fluorescent protein, which is fluorescent only at pH 7. So that means that these receptors, when they're on the outside of the cell, on their way to the synapse, will be fluorescent. Receptors inside the cell in endosomes, which have a pH of 5, are not fluorescent. So as they move from this location to this location, they become fluorescent. You can expose them to X-ray light and photobleach them, and they lose their fluorescence. So here's the experiment. You load up neurons in culture with the fluorescent form of the receptor. So you can see wherever they're on the outside of the cell, they're fluorescing. When they're inside the cell, they're not fluorescent. Then you can photobleach them with UV light, and the fluorescence goes away. It goes away for the ones that are on the outside, but the ones on the inside are not affected by the photobleaching. And then you can just wait, and the intact receptors move up to the membrane, and they accumulate, and then they go to the synapse, and they can actually move out of the synapse and go back down into the cell. So there are several different pools here. There are the receptors, which are extrasynaptic, and they're very mobile. And these are quickly replaced in a FRAP experiment. You'll see that the fluorescence goes away when the cells are illuminated with UV light. And if you wait with time, the fluorescence returns because intact receptors move out to the plasma membrane. So these are quickly replaced. The ones in the synapse are slowly replaced because they're anchored. And this population is also increased by a procedure called chem-LTP, which mimics LTP in vivo. So the experiment involves measuring the recovery from photobleaching if you are wild type and you can be phosphorylated on this critical serine, serine 845, or if you're mutant and you have instead an alanine at this position, and you're still fluorescent. So here's the photobleaching. Here's before exposure to UV. Here's after exposure to UV. The fluorescence is gone. And in each case, it recovers. So the first thing is cells that are in the basal state traffic the wild type and the mutant, here they are, equivalently. So that means that the basal condition, the phosphorylation of serine 845, doesn't make any difference for this recovery. After chem-LTP, the stable receptors, the ones at the synapse, increase. So the recovery, you can see here, the recovery reaches a plateau which is lower than in the basal state. But once again, the mutation of this serine 845 doesn't seem to make any difference. So that means that the receptors accumulate. They don't accumulate because they're delivered to the extrasynaptic membrane more rapidly. Let's look at the alternative, which is endocytosis. Endocytosis involves a complex of proteins called AP2. So here's a receptor in the plasma membrane. It can bind AP2. Once it's done that, it can gather clathrin. It can enter a clathrin-coated pit, and it can be endocytosed and trafficked to early endosomes. Okay. Interestingly, GluA1, shown here, has a sequence, KRMK, just near the plasma membrane, which also appears in GluA2. And that can bind this critical factor, this AP2, can bind at this site. That suggests that this site is involved in taking the receptor off the membrane into the endosome. Okay. So here we compare two groups of neurons, control neurons and neurons that have been treated with chem-LTP. So this is a chemical stimulus that drives the trafficking of the AMPA receptors. And you can see there are two consequences. First, the phosphorylation of this special serine, serine 845, goes way up when the neurons are activated by chem-LTP. Second, the association of the receptor with beta-adaptin goes way down. So this, and you'll recall that beta-adaptin is involved in removing the receptor from the plasma membrane. So this suggests that the phosphorylation, which happens with LTP, blocks the association of AP2. And this would be consistent with stabilizing the receptor in the membrane. So that's represented here. Here is the receptor. Here's the receptor after chem-LTP. Phosphorylated AP2 is released, and that would make it stable. We check this further by examining the sequence that was known in GluA2 to bind AP2, this KRMK. And you could focus down here. This is the binding of AP2 by the wild type, and this is the binding by mutants which alter this sequence. So you can see that the binding is decreased. So that's further evidence that GluA1 binds AP2 at this position. So that result is summarized here. Here's the wild type. If you mutate the KRMK, you don't bind AP2. If you phosphorylate serine 845, you also don't bind AP2. So the conclusion is that this pathway stimulated by sucrose leads to a phosphorylation of the GluA1 C-terminal domain, and this allows the receptor to reside in the membrane on its way to the synapse, and this accounts for synapse strengthening. Okay, so what could be the relationship of these pathways to reward function? Okay, so what I'm going to present to you now is a model which is consistent with the evidence, but is still, you could say, hypothetical. So here is a person consuming something which is rewarding, cereal with sugar on it, and this consumption activates the release of dopamine. Okay? Dopamine is released in striatum, nucleus accumbens, and frontal cortex and hippocampus. Okay? The nucleus accumbens also receives glutamatergic input. You'll recall that this glutamatergic input was required for the receptors to move all the way into the synapse. Okay? So the hypothesis is that these two signals, the signal arising from dopamine and the signal arising from glutamate, cooperate to complete the trafficking and the strengthening of the synapse. Okay? So that's represented here. Dopamine activates PKA. PKA phosphorylates this critical serine, serine 845. That allows the receptors to accumulate, although outside the synapse. Glutamate activates PKC, which allows the receptors to take the second step into the synapse through phosphorylating an additional serine. Okay? So in order for the receptors to function, both of these signals in this model would have to take place. Okay? So let's imagine how this could operate and could provide selectivity for activating medium spiny neurons. So this diagram represents medium spiny neurons, their input from dopamine, and their input from glutamate. And dopamine acts by what's called volume transmission. Its release can affect many neurons. Glutamate is synapse specific, and it would only be those neurons which would receive both dopamine and glutamate which would be activated, and the behavior would be potentiated. I told you that the GLUA1 which enters the synapse is in the form of a GLUA1 homomer. And these are special. As I said, they're calcium permeable and rectifying. The significant point is that work from Marina Wolf shows that these receptor types accumulate in rats which are addicted to cocaine, animals that have cocaine craving. So cocaine addiction and consumption of sucrose have similar consequences for these critical synapses. So you can imagine that the cocaine craving state is related to the sucrose consumption state, in that particular medium spiny neurons are activated. They're constitutively or long-term activated in the case of cocaine craving, and this with cocaine can lead to an addictive behavior. Okay? So what I told you is that sucrose induces AMPA receptor trafficking in medium spiny neuron synapses. The potentiation employs a two-step mechanism. The first involves the accumulation of an extrasynaptic pool through the phosphorylation of this residue, and this is followed by stimulation with glutamate. The two-step trafficking suggests a model for dopamine glutamate signal integration which could lead to behavior selection, and it's relevant to the cocaine craving state. Okay. So I'd just like to thank the people who contributed to this work. And you can see there are many of them, but 3 of them are related to UFF. They're the host today, Jane Ferreira, who was the first person to come from Brazil to my laboratory, Matthias Sattler, who came and then went to Colorado to work with my postdoc, Siga-nos Kim, and is now a permanent resident of the United States, and Matthias's thesis advisor, Dr. Kabrusley, who is presently at UFF. Okay, so thank you very much for the opportunity to present this talk, and I'd be happy to answer any questions. Okay. Thank you. I can't hear you, Marcelo.
Sorry, I was mute. Thank you, Ziff. Thank you a lot for your excellent talk. Always a very exciting and full of data of highest scientific quality. Now we're gonna move on the chat questions, if you have some questions. But I think I perform my questions, okay? Your lab studies a lot of CDK2. Could you give us more information about the role of this assignment on trafficking AMPA receptors in this model, if you receive any participation on this model?
Yeah, so in the pathway I described to you, the first step is the phosphorylation of the receptor by protein kinase A, and that's in response to dopamine. CGK2, the second kinase that you mentioned, can also carry out this phosphorylation. So a cyclic AMP pathway and a cyclic GMP pathway can both drive the trafficking to medium spiny neuron synapses.
Okay. I think Rafael Brito asked a question. Thank you very much for excellent talk. I would like to know if sucrose consumption also increase the number of synapses containing AMPA receptors.
So that would be a separate regulation, and I don't think that's been established. So activity generally increases synapse number, and it's possible that that takes place as well, but we haven't actually measured that. I'm sorry.
Okay. Mariana Wallace Davis. Professor, is this junction of glutamine and dopamine a tool to make a reward system selective to neurons? I don't know.
So I think the answer is yes. So only neurons, of course, would receive these 2 inputs. And what's important about the model is that many neurons would receive dopamine, but glutamate release is synapse specific. So only particular neurons would receive glutamate. Because you need both signals to potentiate the synapse, only the neurons that can receive glutamate and dopamine would be activated, and this would give selectivity.
Okay.
Can I ask a question?
Sure.
Marcelo?
Sure.
So I was wondering how cannabinoids may regulate this scheme. Do you have any ideas about that?
Yeah, so all drugs of abuse trigger the release of dopamine. So cocaine does it by blocking a transporter. Cannabinoids do it by blocking inhibitory neurons, releasing the dopaminergic neurons to be more active. So I would say that cannabinoids feed into this pathway by stimulating dopamine release, which would enhance that first step in the trafficking. So I imagine if you gave a cannabinoid to a rat, you would see the same changes that we saw with sucrose.
Thank you.
Mm-hmm.
There's one more question in the chat. Comes from Cesar. Marcelo, could you ask about NMDA receptor? Have you, Professor Ziff, seen about NMDR in the system? I think yes, no? What
So,
can you talk about?
Almost certainly the NMDA receptor is functioning. And in what I described, it would conduct calcium in response to glutamate. It would carry out the second trafficking step. So it would activate PKC. That would be my prediction.
There's more questions here. Isabella Moraes. Is there any relation of this glutamine or glutamine and dopamine inputs with obesity? Considering an obese person would consume more food sources of sucrose?
Yes, there is a relationship. And in fact, in studies that I didn't present to you, we looked at what happens if a rat consumes sucrose continuously for a long period of time. Of course, it becomes obese. If you take away the sucrose, changes take place at these same synapses. And in fact, what's altered is the affective aspects of the animal. So the animals become depressed when you remove the sucrose, and the neurons that I've been speaking about become less active. So these neurons' activity is correlated with reward. Less sucrose, less reward, depression, and anhedonia.
Really nice. Claudio Serfati. Do you believe that this association of reward inputs changes with aging? What is
I couldn't say about that. It's many things change with aging. But we haven't investigated that. Hi, Claudio.
Claudio is worried about the aging all the time.
Yeah, me too.
Another question from Cesar. How about NMDR interacting with D1 and D2? Thank you.
So I know that the dopamine receptors interact physically with glutamate receptors, but I don't know in this system how such an interaction would change the function.
Okay.
Again, D1 and NMDA receptor could cooperate.
Yes. Paula Campello. Excellent talk. Thank you. The effect of sucrose on the behavior and regulation of AMPA is relatively rapid. Have you tested whether it can be reversible?
No, we haven't. But the studies that I presented show that the potentiation of the synapses lasts probably for at least a day. I imagine the time that the receptors decay, but we haven't looked at that.
Okay, there's more questions here. The chat is Mariana Polis Davis. Professor, is this junction of glutamine and dopamine a tool to make a reward system selective to neurons?
So the the reward system would be selective to neurons, or the other cell type that could be involved would be glia. But it's selective to neurons, and it's selective to particular neurons. That's the hypothesis. And the suggestion from this work is that the actions of glutamate and dopamine together select particular neurons in the medium spiny neuron population. That hasn't been established, but that's what we're hypothesizing.
Can I just ask the last question?
Sure.
I was wondering, like, what kind of stimulus would you expect would pair with this dopaminergic sucrose stimulus from in the glutamate realm? I mean, what kind of stimulus do you think would pair with this stimulus?
I could have explained that better. So the glutamate is coming from prefrontal cortex, and prefrontal cortex, as you know, gathers information of many sorts from different regions of the brain and makes executive decisions. So it could be the environmental component of receiving a reward. So you receive, for example, if you receive a reward in a particular context, then the hippocampus would recognize that context, and it would transmit that information to the prefrontal cortex, which would be involved in decision making, so that the context, or with auditory training, a bell or shock or whatever, could constitute a second signal that would be glutamate dependent.
Thank you. There's still a question from Paula, right?
Yeah. Paula Campelo, did you evaluate any effect on anxiety-like behavior?
So not in the studies that I presented, but in the studies that I mentioned, in which we remove sucrose, certainly anhedonia increased. So animals didn't show after they had been removed from sucrose, they didn't show a preference for sucrose. And on the forced swim test, they became depressed. Whether they became anxious also, we didn't measure, but it's certainly a possibility.
Ziff, another question of mine. Did you ever seen any effect of nitric oxide in this system, especially because CDK2 also, I think, is...
No, but I believe it. I believe that medium spiny neurons do respond to nitric oxide. So it's possible that nitric oxide could replace dopamine, in effect,
okay,
activate CDK2, and could drive the trafficking.
That's my point. That's exactly my point. Okay. I don't know if I have any questions.
No, it's repeated.
Okay. If we don't have... Okay, one more. Yael Abreu Vilaça. Thank you for the talk. Any idea on how would the use of a drug of abuse interfere with a natural reward such as sucrose and vice versa? You understand?
So the question is, how could consuming, say, cocaine affect the action of sucrose? I think people who take drugs of abuse like cocaine, their appetite decreases. So they probably would consume less sucrose. So I would say that there would be an interference by a drug of abuse. The drug of abuse would take over the pathway, and sucrose would no longer have an effect. That's my guess.
Okay. Another question, Rafaela Campagnoli. Thank you for the great talk. Could this enhance AMPA receptors be underlying mechanism for food addiction?
Yes, so people make a comparison between the actions of sucrose, and which would be representative of food, and cocaine, so that it's possible that the mechanisms that lead to cocaine addiction are similar to the ones which lead to obesity. This is complicated by the fact that obesity often reflects what's called binge eating, in which you stop consuming food, you go on a diet, and that makes you especially sensitive to the possibility of eating more food. And when you do that, you consume an enormous quantity. So I would say that the two mechanisms are probably related, yes.
Okay. I think it's finished. Okay. If we don't have any more questions, okay, I will end the chat. Okay. Okay, I will end the chat. Once again, we are incredibly grateful for your participation. Always incredibly good. And okay, there's one more question. The opposite would happen. And the Iael Abreu Vilaça. And the opposite would happen too if you consume lots of sweets. That would decrease the likelihood being addicted to a drug abuse.
So I can't answer that confidently, but you know, it may be that if you have normal pleasures, social interactions, sexual interactions, food, and so forth, that your state of mind doesn't force you to go to drugs of addiction. So it's possible, yes. I don't know how you would test it, but I think it's possible.
Okay, people. Okay. Ziff, we are extremely thankful for your talk and your availability and your presence with us. That we're honored by our event, and thank you a lot. Thank you so much for your presence and your talk.
Thank you.
Thank you.
It's a pleasure and an honor. Thank you.
Thank you very much.
Bye-bye. Thank you.
Bye-bye.
Bye.
Ok, so now... Agora eu acho que a gente vai fazer o nosso intervalo, né? E vamos voltar a que horas?
Acho que é
2 horas. 2 horas. Ok, gente? 2 horas, então tem um tempinho para relaxar e a gente está todo mundo de volta aqui às 2 horas. Até lá.
Maravilha, Cia. Obrigado. Valeu.